what strikes us is that every single person with MS is really quite different to every other person with MS. And in particular, in terms of how the disease unfolds, some people become very sick very quickly, and other people have literally completely normal lives and have sometimes possibly quite active MRI scans showing lots of new inflammation, but it comes and goes without them even knowing about it, and 20 or 30 years later you're still sitting in front of a person who has remained really, really well. So the disease is not, homogeneous. There's not one story for everybody. And we think that some of that relates to just how well or how badly the brain can cope with the extent of the inflammation. Some examples of that include remyelination, so how well the brain actually repairs the myelin that covers the nerves that is damaged in MS. In some cases that's very extensive, in other cases it doesn't seem to happen at all. And another very important example is the consequence of the inflammation to axons. Axons are the cables connecting the nerve cells in the brain, and the cables running down the spinal cord, essentially performing most of the bodily functions. And the problem is that somewhere along evolution, human beings have lost the ability to regenerate those axons. So if you cut one in the brain, if you cut one in the spine, it doesn't grow again. That's different to your arms or legs, actually. If you cut the nerve, an axon in your arm, in the nerves here, it'll actually grow again, eventually finding its target. So peripheral nerve injuries kind of regenerate, but in the central nervous system they don't, which means that every time an axon is killed, that's forever. And it may well be that some brains, some axons, are actually better at defending themselves than others. So these are some of the issues in neurobiology. And then more recently, huge amount of interest in stem cells, which I don't personally work on, but other people in the lab are very involved in stem cell research, and they're trying to understand whether there's an even more profound response in the brains of animals and some people where there's actually even larger regeneration of whole systems of nerve cells produced by stem cells. And the answers to those questions are not in yet, really, certainly for people. But it's not, all of this isn't being studied just for intellectual curiosity or for fun. It's being studied with a very strong desire to understand these mechanisms and to basically make the people who have bad MS have mild MS. So in other words, if we understand why myelin regenerates well in some people but not in others, why the nerves, the axons of some people die and other people survive, then we might be able to turn some of that damage around.
Can you tell us about the development of that genetic research and where it's leading us?
This is a story which is fundamentally driven by technology, actually. So we know that MS is to some extent a genetic disease. In individual families, that doesn't actually happen very much. So the risk of, if you like, transmitting MS to a child is between 1 and 2%. So, on average, to have one with MS, you'd have to have between 50 and 100 kids. So this is a rare event. Even amongst more than 1,000 people that we look after in clinics, many of whom have children, this is a rare event. There's just a handful of families where this has occurred. So at the individual level, it's actually not a big problem in terms of transmitting it as a genetic disease. But at the same time, if we look at people who get MS, it's 3 times as common in women as in men, and that's evidently genetic. And it's also about 30 times more common in Anglo-Saxons as opposed to Japanese or Chinese people, and that's very evidently genetic, because even Vietnamese and Chinese people who've grown up in Australia have a very low risk of developing MS. So, given it's genetic, there's, of course, the desire by researchers to understand the genes. And the technology has moved incredibly quickly. So when we take blood or, in fact, spit from someone with MS or a healthy control, for around $50, 8 years ago, we would have been able to do 50 individual little genetic tests on that sample. For around $50 today, we can do about 250,000 genetic tests. So the amount of data that we're getting is massive. And so this has resulted in massive experiments. In Australia, we have a consortium, well, in Australia and New Zealand, I should say, called the Axons Consortium. It's about 12 centers that have collected DNA from lots of people with MS and lots of healthy people, and that's now also been joined into something called the IMSGC, the International MS Genetics Consortium, who have 10 times as many samples. And the experiment that Jeremy just alluded to will essentially report the results of this kind of genetic analysis in more than 20,000 people with MS and more than 20,000 people who don't have it. This is what we call an association study. We're looking for what genetic variation might be slightly or significantly or importantly associated with MS. So if I take any two of you, you compare your DNA, there's a difference about every 10,000 bases in the genetic code. The genetic code has 3 billion bases. And overall, amongst lots and lots of people, 1 in every 300 of these is different between people. And that's what we're measuring. We're measuring systematic differences in people with MS compared to healthy people, or people who don't have MS. They're not necessarily otherwise healthy, but they don't have MS. And where those experiments are leading us is that we now have identified, we as in the global world of MS has now identified about 100 of these. And these are not directly disease-causing genes. It's more like, I like to say, it's more like blue eyes versus green eyes. So blue eyes, slightly more common in MS, green eyes, slightly more common in people who don't have MS. It's that kind of information. And what it actually represents is just the first layer of an onion. You know, onions have lots of layers, and we've opened up the first one, and that's actually good news. It tells us about a really complex puzzle as to what might cause MS. But there's about 19 layers to go. Every research institute whose websites you might peruse will tell you about bench to bedside. And the catchphrase is really that we can do lots of really very wonderful and elegant experiments in mice, and there's a massive amount of value and understanding to be gained from that. But there can be a tendency for that to be a means to an end, to kind of create more beautiful research in mice. Obviously, the focus of all of that basic research, when you call yourself the MS group, is to ultimately translate that into treatments for people with MS. And the focus of the Florey Institute and the University of Melbourne in neurosciences, and MS is a part of that neuroscience focus, is to find ways of actually going from bench to bedside and not just to say it. So about two buildings to the north here is a building called the Melbourne Brain Centre, which is a neuroscience centre which will combine the new Florey, the Mental Health Research Institute, and quite a lot of the neurological research of the University of Melbourne in one place. But also, there's a mirror centre in 2 hospitals, a large one at the Austin Hospital and a smaller one called the Melbourne Brain Centre at the Royal Melbourne Hospital, which is being built across the road at the Royal Melbourne Hospital. And you know, in the past, unfortunately, that big road, Royal Parade, between a huge clinical centre and a huge research centre, i.e. the university, has not been kind of crossed all that often. It's been done well in some areas of cancer particularly, but overall the translation has been minimal. So the idea is really to connect that connect the two sides of that road physically, even down to the same color scheme, so that the researchers can feel more comfortable walking into the hospital and recognize a bit of home. And most importantly, that facility is going to be right next to the clinical trial facilities, where the research nurses are working on clinical trials with people with MS. Immediately on the other side is the ward where people with MS come to be treated. So that kind of physical location is very exciting, and the vision for that part of the Melbourne Brain Centre is to specifically translate from bench to bedside for researchers. So that really means conducting early phase clinical trials involving imaging capacity, blood markers, new drugs which are being developed, and to allow researchers and patients to interact in that space. So that's been funded by the federal government and had operational money, about $3 million from the federal government, had operational money provided to it by the National Health and Medical Research Centre, about $500,000 a year for 5 years, recurrent funding. So that's quite a substantial pot of money to actually build something, and we're very excited about that opportunity.